Okay, we're going to collect some water samples for the Watershed Rules of Life project. I've got one of the brown bottles that we normally use. There's a bunch more sitting here. And this is the nutrients bottle for the whole water. Right, so this one is unfiltered water. So we have to find a spot in the stream that's deep enough that I'm not going to bump the bottom with my bottle. And in this case, it's right out here. So you dip the mouth below the surface. Get some water. And dump over to the side so you're not dumping back to the same place where you're going to be filling again. That's 2. That's 3. And now I'm going to fill it all the way up. Again, don't bump the bottom. And make sure you fill below the surface. You don't want it to skim off the surface. You want to have the mouth entirely submerged. All right, I'm going to collect the rest of these now. All right, so I'm going to fill the nutrients, whole water, unfiltered nutrient bottle. You can use the same technique I used for the big brown bottles, rinsing it 3 times, submerging below the surface. And then these are going to be frozen, so you want it, that's a little bit empty, hold on. Want it down to the shoulder, see that? Mm-hmm. So that is room for freezing. And then seal it up, and that's it. Okay, now we're going to filter the water samples we just collected. We're going to filter them for DNA and RNA. And we're going to collect some nutrients and dissolved organic matter. So I want to show you how we set everything up. We just got back with our bottles. We got 4 brown bottles here. And I'm going to set up the pump for the DNA and RNA samples. This one is a 3-headed pump. You may have a 1-headed pump. Either way, it still works fine. This is our Y tubing for collecting 2 filters at a time. And on one end is our sterile dipstick, which is really just a pipette, where we broke the tip off. You can kind of see that in there, and took the little cotton plug out. Got to take the cotton plug out, doesn't work without it, and then stuck it in there. And then you have this nice sterile dipstick that's in this sterile sleeve, ready to go. Okay, and on this end is where we attach our filters. So I'm going to set up the pump with this and show you how to set it up. I like to have this hang just about at that level. And then within these things, these are the pump heads. This fits into the pump head like that, and you pinch it down. And if you look right here, you have to be careful not to pinch the pump like that, because that kind of seals it. They've got to be in the right groove there, so that it won't pinch the tubing closed in the wrong way. I'm going to set up the other one. We're doing 2 at a time. Okay, so those are ready. And you see these things are flopping around, but they're in here, so they're staying clean, right? Got to keep these things clean because they're going to dip right into the samples. These are how we measure the volume that goes through the filter with these towers. There. You see how they kind of, the height is just about right, so the tubes can go into the graduated cylinders. That's what they're called. Okay, that's ready. Now, just to the side, I wanted to say, this is the whole water nutrient bottle that we just collected. I'm going to go put that in the cooler. So let's go do that. Lunch. So this is an ice cooler, and you can see all the samples we've collected so far today and yesterday. Nutrients and DOC go on ice. I think everything else goes in the freezer, this is dry ice. We'll get to that later. Okay, so when we start, when we get ready for these trips, we make sample packs, and you'll probably do the same thing. We've already pulled a few things out of our sample pack. This is the one for Whychus Creek at Sisters. And these are the 2 filtered nutrients and DOC bottles. And here's our Stervex filters for DOC. And there's a couple other things in there we'll get to. So there's the Stervex filters for our samples. And we're also going to fill these with water that goes through those Stervex filters. So let's do that now. Right, okay. We're going to use a couple of these bottles to start filtering. Give them a mix. Make sure the particles are resuspended. Come up. And then sterile dipstick, or clean dipstick, I guess. Just do that. Save these. Don't lose these. We'll put them back in there after we clean them off. Now I'm going to take these out of here and give the tubing a rinse. There might be a little sample left over from the last run. So I'll turn this on and draw some water up. Now if you come up here and look at the pump, let's see, I have the pump set not all the way high, just getting kind of close. And this controls the direction of flow. And you'll know quickly if you're going the wrong way. And now you can see water's coming out of the ends of these tubes. That means it's been rinsed, so we're good there. Now I'm going to attach Stervex filters. These things are, we've got them labeled A, B, C, D. There's A and B. Alright, so to stay organized, I put A all the way on the left and D all the way on the right. You know, left to right. So let me see. This one, so A, twists right on there. B, twist right on there, and it's just hand tight. It doesn't even have to be particularly tight, just hand tight. Okay, so A, B, there. Let me get CD set up. We use these little Stervex filters, which cost about $4 each, because the filter is kind of protected, and there's little chance of contaminating it with your hands. But you don't want to like dig your finger into anywhere. Just use gloves. Keep your hands clean. All right, C and D. So I'm going to get these started. It takes a little while to filter. You don't want it to take too long, but you get it going. So come down here and take a look at these things. You can see there's water going through. There's no big air bubbles here, which is nice. Sometimes you get air bubbles in there, and when you do, you just stop the pump, and the air will go out this little air valve right there, and then you start it back up again. You see, I actually got rid of some of the air that was in there. So those are running. So those are going to run for a while, and we use these to keep track of the volume. If it fills up, you stop the pump, record the volume, and dump it. Alright, so while these are running, I'm going to filter these things. This is the 0.22 micron pre-filtered nutrients. So these bottles have to be triple rinsed, just like everything gets triple rinsed. Put that cap up like that so I don't get dirt in it. Put a little bit in. Now remember, we're trying to keep track of the volume that goes through this filter. So I'm going to pour this back in here, right? For the rinse water. That's pretty important, because if you're using this rinse water, you don't really know the volume. It's inaccurate. And if you just dump it over here, then you've reduced the actual volume that you're recording, and it'll be not accurate. So that's 3. Alright, so 3 rinses. Now I'm going to fill this thing. And this thing holds 125 mils. So I do know how much I'm taking out of this graduated cylinder when I fill this. Some of you, the samples that are being collected near the mouths of the rivers have a lot of stuff in them, so they'll clog pretty fast. So using this technique, you got to do it right away. Otherwise, these Stervex filters get clogged, and it takes forever to fill these bottles. The site we're at right now, the water's super clear, so we're going to be able to put a lot of water through these Stervex filters. And again, right up to the shoulder. Maybe a touch. I'll do the other one. I'm going to take them both out of this one. They're still running pretty fast. So 250 mils out of this one. So when I record this volume, I'll record that 250 mils as well. It's starting to slow down a little bit, so it's not filling as quickly. Okay, so these two go in the cooler. I should point out that the other one is the DOM, dissolved organic matter, 0.22 micron pre-filtered. So there's 3 of those bottles per site. 2 filtered, 1 unfiltered. Alright, what do I do next here? Wait till those fill. Alright, I stopped the pump because these things are getting full and I have to make a recording of the volume. So you can see that the filter, they can sit down in the water like that, that's okay. But to measure the volume, you got to take it out. So you take it out of there and then pick it up and try to level it and get the volume. So that's a little under 990 mils. So that looks like, I'm going to record that as 990 mils. So that's A. I'm writing it on here because Ted has the clipboard, but I'll record it on the clipboard when he gets back. 990. Well, and then you can dump it out because we got to keep going. It's not the filter isn't clogged yet, so we're going to keep filtering. 80. 70. And this one is at 725. And this one is where we took out that extra 250 mils, so I'm going to put that on here too, plus 250. Let's see. Dump it, and I'm going to keep going. So we'll keep filtering. Like this. A, B, C, D. Alright, so the Stereovex filters are getting full. You can see that they're starting to drip at the ends. That's actually pretty doggone full. It's not streaming out anymore. You can stop a little before it gets to this point, but we're going to stop now. So up here, what you do is you take the dipsticks out of the samples and kind of carefully flip them up like this and just stand there for a while. And what's going to happen is the pump is going to pull all the water through out of the tubing through the filters, and it's going to push all the water out of the filters so they'll be dry, or moist, I guess. No water in there. The reason I'm holding these is to keep them clean. Can't put them down. Still dripping out. There you go. See, it's pushing all the water out of there. And it starts to slow down at this point because some air is getting through the filter. Sometimes I'll just crank up the pump a little bit at this point, all the way to high. Here we go. That'll help push the last bit out. we clogged these filters pretty hard that time, so that's why it's not emptying very quickly. You can still see there's a little dribble in the bottom there. Dribble of water. So then if you leave that water in there, all it's going to do is dilute our buffer and make it not as effective. So we want to get the water out. Sometimes you can do this, give a shake, get it out. That looks pretty good to me. What do you think, Natasha?
Yeah.
I see one little drip in there that's never going to come out. And then that's it. Okay, so those are done. Stop that. So I stopped the pump. I'm going to put these sticks back in the water here so they don't get dirty. And just leave them there. So now we've got to take these off and seal them up and put the fixative in. So I'm going to start with these two here, which look good. So take it off like that. Screw it off like that. Let me get all four. Okay, now we have a kit of things for killing. Let me pull all that stuff out so you can see it. One of them is sealant for the out port. These are caps for the in port. We've got our RNA later syringe, which I haven't labeled. I should have labeled. And it looks like that's getting empty, so I'm going to have to refill that. Here's some more RNA later. And I think that's everything I need. Okay, so these things, that little nipple there at the end needs to get sealed with this putty here. So let me see. I like to dip it in twice, so I go in once and then a second time. Okay, let me show you. So you can see it fills that end cap there. Do another one. Oh, I didn't get any on that one. I got a little bit on that one. I'll put a little more on. Okay. I have to refill this syringe. There's a little bit in there, but not enough. So I'm going to take this cap off, this syringe filter off, and pull the plunger out just past the crack. You'll hear the crack. Yeah, there you go. But I didn't pull it all the way out because I don't want to spill that. Put that back on here. I can work that out. I like to hold it like this to keep that rubber part clean. Good, things aren't working perfectly. So you can see in here that there are some crystals. So it's not fully dissolved. It's because it's cold outside. Can you see them on the video? Yeah, okay. Yeah, so anyway, I'm gonna warm these up on my hand before I use it. But so if you flip it up like this, you know, and kind of try to make sure this doesn't pop out, take this out and get the air bubble out. These syringes all have a point where they have to crack past. I don't know, there's some sort of thing. So sometimes I spray it in the air. It's not good to do that. So I'm trying to keep control over it. Oop, there we go. Ready to go. So I gotta warm that up a little bit. I don't know if that number of crystals is that much of a problem. I don't think so. That many crystals is not a big deal. If it's like cloudy white, you got a problem and you've got to get it warmed up so those things dissolve. So on the end of this we keep a little lure plug, thick lure plug, so that it doesn't drip all over the place. So I take that off, take one of these, put it on there, see it just screws right on, and fill it all the way with this liquid. All right. One. Now this stuff is called RNA later, and what it does is it diffuses into the cells and stops all the enzymes inside the cells from working. And by doing that, it keeps the enzymes from destroying all the DNA and the RNA so that we can extract it later. And that's why it's important to let these incubate for a little while. So after I fill these and seal them up, we're gonna store them in the dark for like 10-15 minutes or so. And that gives it enough time to get into the cells and preserve the DNA and RNA. So I close that back up so it doesn't leak. Now we have our caps. My fingers are big. One, two, and by doing it that way, I didn't contaminate the rest of them. So these are ready to go, and then you've been using this technique, right? So one way to preserve them, you can put them in your pocket, but maybe they're wet or something. You can just put them in your glove for a little while, and then we put them over by the cooler for 10-15 minutes before we freeze them. All right, one of the important measurements that we make are cell counts. We count the number of microbes in our samples. And in order to prepare the sample for that, we have to fix or kill a sample with paraformaldehyde, right? So we have to measure that into a tube. And one of the tubes that you're using is for that. We've been using these green capped 5 milliliter tubes. See, it goes up to 5. So we have to put 3½ milliliters of water in here. And I do that with a pipette and a bulb. You use these bulbs before. Okay, so I think we're shipping you some of these. This is a 5 milliliter pipette, and it's packaged so it's sterile, clean. We don't have to worry about any contamination. So you open the end that has the cotton plug on it and squeeze your bulb onto it like that. Now the bulb is supposed to create a vacuum and suck liquid up into the tube. So if you just pinch the bulb, it creates the vacuum. So that's ready to go. That pushed a little air past this white thing. And then to suck up, we'll squeeze this button here. Okay. So I'm going to take it out and dip it into the sample. And suck up some liquid. And if you do it too hard, you'll suck water up into the cotton plug, and then you have to throw it away and start again. So I'm just pinching a little bit at a time. I have way more than I need right now. So over the ground, I'm going to drop it down to, where's my measurement? 1, 2, 3, 3½ mils. So I'm gonna, I better show them this. This button draws up, this button lets out. So it lets air in here and it can fall out. So I'm gonna bring it down to 3½. Now I've got to open this without dripping anything. Put it in there. And then after you finish draining it, you can squeeze this little this thing here, and that pushes the last little drip out, which is satisfying. So that's ready to go. We have not been reusing these. We use them for one sample and throw them away. Okay, so now we've got the sample in there, 3½ mils. Now we have to add the paraformaldehyde. And that paraformaldehyde we store frozen, but you have to thaw it and get it dissolved again before you put it in here. So I keep the fixative over here so it's out of the way. All right, so I'm finding that I keep the paraformaldehyde over here to keep it out of the way. Here it is. It says 10% paraformaldehyde made in September 2019. And I have to measure 875 microliters of this into here so that the concentration comes out correct. So you'll have a P1000 pipette, 1000 microliters, that's 1 milliliter. And I've got it set up for 875 microliters right now. Pipette tips that fit the pipette. Get one of these. Push that down on there. So, you know, when you use these things, they have 2 stops. They have one stop here and the second stop there. So you'll feel it when you use it. That first stop is the measuring stop. So you go to the first stop to draw up the volume that you want, right? 875 microliters. So I'm going to carefully open my paraformaldehyde tube and kind of hold that there and take this down to the first stop and then dip it in and slowly draw it up. All right, there's the volume that I need. Like this. Open this one and just squirt it right in there. And that second stop is really to blast the last little drip out. That's all that's for. Second stop on this thing. Okay, so now everything's sealed up and I give it a mix and then I freeze that. Well, I'll show you where I do that. So put that down. Now this has a little bit of paraformaldehyde on it, so you want to be careful with that. Don't just rub it on anything. Put it in the garbage. So we have other samples in here. I'm going to put it with these. And it goes in the dry ice container to freeze. So you can see all the other stuff we have in there, the big blocks of dry ice.